Hello, the large family of MD Anderson. It's 10 a.m. Houston time. It's Enjoy Science time. And I want to thank you very much to all of you from here, Houston, and from all over the world being with us because of your support and because of so many wonderful talks by our speakers and because of the support from the institution. We decide to continue Enjoy Science for the full summer, and we are waiting for you to be with us all summer long. Now, as I told you all the time, Enjoy Science, it's because of you, and it's also because of the wonderful clinicians, scientists, and our patient at MD Anderson. It's a webinar that I host with my friend and colleague Alessandra Ferraioli. It's under the Office of Chief Scientific Officer, and Dr. Giulio Draeta give us all the support for having this done at the best. And of course, we have to thank Ellison Gardlis, Kent, and Valerie Jimenez, because without their dedicated work, this could not be done. And please use the chat function for question, and please go as frequently as possible to the webpage of Enjoy Science to see previous talks, because these were really wonderful talks because of the wonderful speakers that we had. And in fact, today we have the pleasure to have Dr. Phil Jones, our speaker. And why it's a big pleasure? Because Phil is bringing in MD Anderson with a full team of IACs. As you know, he is the head of drug discovery in the Institute of Applied Cancer Science. It's also the vice president of therapeutic discoveries. They are bringing an expertise very much needed in an institution like MD Anderson. The expertise of how to prepare and how to have drugs done at the best for clinical trials and for our patients. And I know Phil for a long time, I know his team for a long time, simply because I collaborate with them and simply because Ajax is a few feet away from my actual office. And I can tell you that these are very hardworking people doing the job at the best and trying to combine very much our research strength with their wonderful strength of applying the drugs and developing the drugs as the best. And who is basically the leading person and the person behind all this wonderful effort? It's Phil Jones, doing work day and night. I saw him early mornings when I go at work. I saw him late when I leave the work. He's still there doing the job. So I'm pretty sure today we will, all of us, learn something that really many of us, including myself, are missing. How really these drugs are developing well for clinics, as Phil has a wonderful experience being previously in big companies, having experience in how these things are done in companies. So thank you, Phil, for your time to be with us, and we are very happy and very curious for your presentation today. Good luck.
Well, thanks for that kind introduction, George. And now I've got a tough act to follow here. So just a reminder to everybody who's on the WebEx here, please use the chat function here for your questions and answers. And just to highlight the facts for the next week, we've got Katie Rizvani coming along to talk as the next part of this symposium, which will fit very nicely following on from this presentation. So today for the talk, I really want to talk a little bit about some of the adventures in oncology drug discovery that we've been doing within the Therapeutics Discovery and Development Group. And really focus on one of the programs we disclosed for the first time back in April for a first-time disclosure at the AACR meeting. That being the development of agglutaminase 1 inhibitor, the molecule that was formerly known as IX6274 and now goes by the code number IPN 690. So for those of you at MD Anderson, you know that back in 2011, we decided to start conducting an experiment. What would happen if we were to move away from a traditional relay race of drug discovery, whereby discoveries are made in labs such as we have across the campus, industry then develops a therapeutic, and then they get passed back to comprehensive cancer centers to conduct clinical trials. What would happen if you were to put all of those components together uniquely at MD Anderson? we really thought that this would be give us a huge opportunity at the institution to develop impactful therapies. And I hope in the next 15 minutes or so that I'm going to really prove to you that we've been able to fulfill that mission. Again, what is now known as the Therapeutics Discovery and Development Group started in November 2011, and it has continued to evolve and will continue to evolve as we move into this decade. We really thought this would be a new framework for developing both therapeutics, potentially for diagnostic tests, and really to help stimulate the external environment here. And it really creates novel therapeutic opportunities for our patients here at Anderson and beyond. And it also offers the opportunity to create long-term sustainability for the organization. We started off with the small molecule efforts. We've then expanded to enhance our translational biology capabilities, forming the Traction platform that's led by Tim Heffernan. We have expanded and built a neurodegeneration consortium that reaches outside of Houston up into the Northeast, led by Jim Ray here. We've enhanced our biologics development capabilities and created the Orbit platform that's led by Dongjing Zha. And more recently, we've really expanded into our biologics development and now cellular therapeutics development led by Jason Bock with the acquisition of our Bellicum CTMF facility earlier this year. So all of these 5 platforms now are really created to eliminate some of the bottlenecks that traditionally hamper therapeutic discovery and are really focused around developing therapeutics inspired by the needs of our patients and guided by our clinical faculty here. We're really trying to create life-saving, transformative medicines for our patients here. And we believe that within the framework of MD Anderson, collaborating across the institution, we should be able to do this quickly, safely, and effectively, and really be able to drive our therapeutics at those areas of unmet medical need, and really to target some of the patient populations that are currently underserved by some of the standard of care agents and some of the agents that are currently in development. So where else on this planet is it really possible to come from an idea and transform that into a response in a late-stage cancer patient? That's what we are really hoping to be able to do here within the Therapeutics Discovery Group, to take the ideas that are coming from our clinicians, that are coming from the faculty and the other researchers that are on this WebEx here, and really to carry those ideas and create transformative medicines here. And it's not just us leaders that are doing this. The Therapeutics Discovery and Development Group now is around 150 researchers that come from a real cross-function of experiences that have come across the globe to join the organization. And the goal here is to develop a continuous flow of clinical candidates, really with strong translational data and hypotheses here, both small molecules, biologics, and cellular therapies, and get those into the hands of our clinicians, be able to treat our patients here, and to be seeing responses here. And that we really leverage the concept of project teams to move our projects ahead, and we're bringing in that multidisciplinary skills that we need to move our projects down the development pipeline. And it's kind of the strength of the collaborations that we have across the institution. This slide just highlights a small number of those. We are really collaborating with most of the lab research departments here. We're collaborating with most now of the clinical departments. And this really enables us to get our projects into those areas of unmet medical need and where we think they are going to be really most beneficial here. And I really thank all the collaborators that you can see on this slide and those of you that I've left out here, because without these collaborations, it would not be possible for us to be so successful. So when we start thinking about any drug discovery project, we really think about four key aspects here. The first really being the disease relevance. Can we show a true dependency around a particular therapeutic target here? The second is really around the clinical need. Do we think that there is actually a need for an additional therapy in that particular indication? Do we think that this is something here that would really target those underserved patient populations? Clearly, we need to be successful, so we do think about something here about the ease of being able to develop such a reagent. And our portfolio is evolving. We're moving more and more into innovative space, and with the expansions into biologics and cellular therapies here, we can go into areas that we have previously not been able to go to. And we also think about issues here, kind of what would be the therapeutic index here, how many other people are working on this program. So when an investigator approaches us with an idea, we sit down with him and her and really we have the concept of a target CV here that attack all of these four aspects. We will evaluate that, and quite often we'll turn back to the investigator and say, go around a couple more experiments here and come to us in 3-6 months when you've got that data. But the hope here is that we're really developing a compelling story, and we decide to join forces and collaborate and move forward in this way. When we do so, we commit significant resources, and you'll see the example of this in the glutaminase project. Just the scale that we actually have to prosecute these programs to make them successful. We develop a work plan here. We really put key stage gates in here and thinking about what is that next killer experiment going to be. And we march down the drug discovery continuum and hope that these projects will be successful. Since we initiated the division, we've now been able to put 4 programs into the clinic. The agonistic OX40 immune checkpoint here is now seeing several hundred patients. GSK have this program now in late-stage clinical development, and that one's kind of the furthest down the pipeline. The next 2 programs you'll see on the slide here in the area of cancer metabolism. We've told you many times before about our OX40 inhibitor, and the dependencies that we've been able to show both in leukemias and in certain solid tumors here. And that project has now seen more than 50 patients or so in the clinic. I'm going to tell you now about our second cancer metabolism inhibitor, our glutaminase 1 program, and that is currently dose escalating. We're pretty close to recommended phase 1 dose in the clinic, led by Tim Yap. And then our anti-PR1 HLA2 antibody in partnership with Astellas now is dose escalating in the leukemia group. So 4 projects already moving forward in clinical development. And just on our small molecule pipeline, we've got a substantial number of projects moving along rapidly behind here. Indeed, there are actually 4 projects here, our SHP2 inhibitor, our ATR inhibitor, a neuroprotective agent, and a YAP1 inhibitor. They should all be able to move into first-in-human studies in the next 6 months here. Behind that, we are invested in the areas of epigenetics with 2 projects here that we hope to be pushing into development. We continue our efforts around other neuroprotection and neuroinflammation targets here. And there's much more in the background here that I just can't fit on this slide here. But all of these are really focused around areas of unmet medical needs, and we hope these are going to be successful in our patients. So we've been in existence for 8 years. We hit all of our 5-year objectives here. We're now really focusing on how to continue to transform the therapeutics discovery groups here. We hope within the 10-year frame we'll have 1 program in registration trials here. We hope we're going to have several programs demonstrating clinical proof of concept in distinct patient populations here. We are expecting that PRMT1 is the first in-house program to advance into clinical trials. I think, as you will see on that previous slide, with the number of alliances we've got, we have a global reputation for innovative cancer drug discovery, translation, and clinical development. And from the strength of our collaboration, I think we've got a really good cross-fertilization going across the organization here. So with that overview, really let me dive down into a program and give you an idea of how we operate within the therapeutic discovery and development groups here. As has been mentioned before, we've really focused some of our initial programs in the area of cancer metabolism. We thought back in the day that really this plays to some of MD Anderson's strengths. We really have the opportunity here to learn around cancer metabolism from what is going on in patient samples here. Tapping into some of the core facilities in the metabolomics group with Phil Lorenzi leading and others, we really had the opportunity to understand some dependencies here and be able to position some of our cancer metabolism programs in distinct patient populations and combine those effectively in clinical trials. And you'll see from this program that we have been able to do so. So glutamine is clearly a very abundant metabolite in circulation, and cancer cells hijack this metabolite here to really fuel a number of biosynthetic processes here. Glutamine gets processed by glutaminase 1 and glutaminase 2 to glutamate that then can kind of get fed itself into proteins. It comes in here to fuel the TCA cycle and generates a number of key building blocks in cells, including glutathione, including nucleotides. And we really thought that we would be able to identify distinct patient populations that respond to glutaminase inhibition, and also to be able to exploit adaptive responses to really think about combinations here. And I'm going to show you some data there. Besides the tumor intrinsic activity, whereby glutamine gets taken up by distinct cancer cells to fuel the TCA cycle to generate those cellular building blocks, it also has an impact there on extrinsic pathways. And when these cancer cells are there acting as a sink for glutamine, it is essentially depriving all the immune cells here of glutamine that they need for their function. And we hypothesized, and we have data now to support, that if you basically inhibit glutaminolysis within cancer cells here, you restore the pool of plasma glutamine and make that available for immune effectors, and really you're also being able to drive now an immune response in our patients. And that's clearly playing into some of our opportunities here for clinical positioning. So behind any program, there are really three key attributes to success here. The first of is really around, can you understand the disease biology? How do we actually really understand how glutaminase 1, glutaminolysis impacts on the maintenance of tumor cells, of the tumor microenvironment there, and how we're going to actually be able to position this program? The second key piece here is really around the quality of the therapeutic we develop. Ultimately, this needs to get into hundreds, if not thousands, of human patients. It needs to be effective in these patients. So how do we engineer that one molecule that actually has all the right attributes so that it can succeed in our cancer patients? And the third one is really being able to demonstrate that this works in the clinic. It is great if you can have the first two pieces, but you will fail if your medical oncologists here are unable to demonstrate that the program to get the right patients to enroll, to show responses in those patients. So these three pieces are fundamental to the success of any project here. To bring it together, as previously mentioned, really requires the work of a large team. One individual on their own is going to be unable to succeed here. To solve those three puzzles, it really needs a good cross-sectional group that comes together to make that successful. And that is what we were able to do with the glutaminase 1 project. I really want to acknowledge up front that what I'm going to show you in the next 30 slides or so has really been the work of this project team on the left-hand side. Probably around 50 or so of the people within the IX and Traction group, led by Jeff Kovacs here on the translation side, and Mick Soff on the drug discovery side, have made this project successful and got this into the clinic now that Tim Yap and his colleagues in ICT are able to demonstrate that this program is working. It's not just the work that's been able to be done within the therapeutics discovery group here, but it's the fact that we've been able to be tapping into the metabolomics group here. It's the way that we've been able to work with the pathology and lab medicine group here with Jing Song and his team here, and Billy and Russell, to really develop our CLIA-based IHC assays. We've been working very closely, as you'll see, with the thoracic group and the gyn-onc group here around our translational hypotheses here, and working again on some of our patient profiling now with TMP and IPCT as we're thinking about enrolling the right patients and profiling some of the correlatives that come back. And now Ibsen with the global project team here are providing clinical support. So everybody mentioned on this slide, and the others that I've obviously missed from this slide, and I apologize, have made this project successful. So from a drug discovery standpoint, we were interested in inhabiting this enzyme, glutaminase 1. So glutaminase processes glutamine, it creates glutamate, and it exists as a tetramer. Each of these units is one monomer here. There is the active site in each of these monomers here. And we knew from the literature that a series of molecules acted as allosteric inhibitors. They pull this particular flap out of the active site of the enzyme, and by binding these small molecules, they lock the enzyme in an active conformation. And it was really, this was the building blocks from our project. We knew that these molecules make a number of key interactions with the protein, engaging leucine here, 323, phenylalanine 322 here, and lock the conformation in this inactive form here. The problem with the molecules that are out there, either these cell tools, BP tests, or the Califera molecule that is currently in the clinic, is these are suboptimal. They really have a number of flaws in them that do not enable these molecules to effectively inhibit glutaminase 1 in patients. So we really wanted to come along with a best-in-class molecule that had that right blend of properties. It had the right potency, it had the physicochemical properties and the pharmacokinetics, so that we could inhibit glutaminase 1 effectively 24/7 in a large subset of patients. And we recognized that to be able to do so, we didn't necessarily need the most potent compounds here to enable us to succeed there. So to enable us to get there, we set up what we know as a screening funnel. We really think about kind of building the set of assays that are going to help us here to find that key molecule that's going to be effective inhibiting glutaminase 1 in patients. So we start setting up an in vitro pharmacology group here, set up our biochemical assay so that we could screen glutaminase 1 in high throughputs, hundreds of compounds a week here, and get nice dose response curves. We set up additional biophysical assays. We had co-crystal structures that were able to drive our projects moving forward. It's great that we can inhibit the targets in vitro and a recombinant enzyme. We also wanted to be able to show that we could to do that in cells. So we developed a target engagement assay where we could monitor the conversion of glutamine to glutamate and our ability to inhibit that. We also set up some really nice flux assays and additional metabolomics that we could really demonstrate here that we were able to inhibit glutaminolysis in these A549 and other cells. We also wanted to demonstrate phenotypic assays, so we set up matched pairs here where we were able to screen on the A549 cell line here, demonstrate that we were able to see a phenotype in our non-responder cells. There was no impact there. So this really helped us to show that we were seeing the pharmacology that we wanted to. We need to think about selectivity. There is a glutaminase 2. We really didn't want to be inhibiting that particular form of glutaminase, so we set up a counter screen to demonstrate that was we weren't inhibiting that. There are another 19,000 proteins in the human body. We wanted to know that we had selectivity and we weren't having off-target activities here. We need to engineer the pharmacokinetic properties adequately so we can get that 24/7 coverage in patients here. And ultimately that we can demonstrate responses in our preclinical models and really have an understanding that you're inhibiting targets off of a certain plasma concentration and showing efficacy. And ultimately showing these molecules are safe here. And so the project team really came together to make this happen. So coming back to BP tests, the molecule, we really thought about how we were going to evolve this so that it had those right blend of properties to be successful in patients. I've just color-coded here on this slide 3 portions of the molecule. The first piece was really around the core here. The ones that the residues here that are engaging that lysine, that leucine, and that phenylalanine here. We wanted to do something around this flexible linker in the center of the molecule here to give us better pharmaceutics property. And then we wanted to change the extremities of the molecule, these wings as we're calling it, because these helped us to give us our molecules the overall blend of properties that we wanted. So all of our work really starts from computational design. We really leverage our knowledge of the structure, working closely with our computational chemists. They design molecules that we think are going to be able to effectively inhibit our glutaminase 1 here. We then really get into the lab, and the medicinal chemistry group get involved in molecular cookery. So they take bulk chemicals coming from a petrochemical industry. They carry out distinct cookery steps in the lab so that they can kind of go from those essentially raw materials to develop a number of compounds each week. And for a full-blown drug discovery program, our teams could be making so around 50 new molecules every week, and then coming in and profiling them in some of the assays that I showed you on the previous slide. Doing this at the onset of the project, we were rapidly able to make around 30 distinct series of molecules. And so you can see in the blue colors on these figures that we've made subtle changes to the central cores of our molecules here. One series here having this bicycle, a second series having this pyridinone scaffold, and then these 5-membered rings here. We profile these molecules both in our biochemical assays here along the x-axis, and so going from a low nanomolar concentration to a couple of 100 nanomolar here on this plot, and then for their ability to inhibit glutaminase in cells. And unfortunately, for the first series of compounds, whilst we could get good biochemical compounds, we weren't able to effectively inhibit glutaminolysis in cells, so we called a no-go here. For the second series of compounds, it was great that we were able to actually get compounds that had good cellular activity, but ultimately these molecules had poor stabilities. Fortunately for us, on this last series of compounds here, we were able to get compounds that started to have the profile that we wanted, and we started to get excited and thought that we may actually be able to be successful with this project. So the next stage here, it's great that we can inhibit glutaminolysis in cells, in plastic, but how do we really need to think about what happens in the human body? So every time we take an oral medicine, the first thing that needs to happen is it needs to dissolve in the stomach. It then needs to get absorbed across the GI tract here. And then the first thing it's going to see is it's going to see the liver here. And the liver's job is to really eat up foreign molecules and degrade them. We need our molecules to be stable in the liver to survive that metabolism and get into circulation in the body, and then distribute into tumors, ultimately cross the membranes to get into those particular cancer cells here, zone in on glutaminase here within the target, within the cells here, shut it down effectively, and then ultimately kill it. So just from taking a medicine, there's a lot of things here that we need to be able to accomplish to get our glutaminase inhibitor to be effective in killing those cells. So we're running here a number of assays, again, coming back to these 2D plots. And in this case, I'm still showing up on the y-axis here our ability to kill the A549 cell lines. But now I'm plotting out against solubility here on the left-hand side of the slide. And we wanted our molecules here to have good solubility in media here. We want them to be able to dissolve in the stomach because you need a molecule dissolved for it to be absorbed. Over on the right-hand side here, I'm showing here a plot, again, of our A549 cell killing versus our stability in liver microsomes. And we wanted our molecules here to have good stability. We want them to remain in circulation. We don't want them to get degraded here by the liver in circulation. So we're continually, the project team is thinking about how do we get into these beige boxes on these plots here. So early on, they were able to identify this molecule here that goes by the code number 12829. That's starting to get where we want it to be. I mean, it had good activity on the enzyme. It had good activity in cells, which also is an indicator that these compounds are permeable. And it had good stability in both in liver microsomes, but also when we gave the compound IV to rats here. The big issue that we had with this molecule is that it had no oral bioavailability. And for those of you that, just to explain that in a little bit more detail, that basically means that 0% of the dose that we are giving, in this case to rats, actually is able to get from dosing orally into circulation. So this clearly shows that we have issues here. And we're not able to kind of be able to dose these molecules orally in patients. And we recognized here that probably it was the solubility of these molecules that was the rate limiting step here. So the project team went back to the drawing board here. They're continuing to beaver away in the labs. They're continuing to make molecules here on a frequent basis. Those molecules are getting pushed to the in vitro pharmacology group that are evaluating them in our automated assays, really looking for potency, efficacy, selectivity. In parallel here, the ADME PK team is really now screening them for metabolism, for protein binding, for solubility, leveraging a lot of our mass spec technologies here. And with that, they're kind of pushing back information in real time on a weekly cycle here. The project team is going in here, is analyzing this wealth of data that is coming out and really understanding that those changes, those tweaks that they've made to their molecules, what are they going to do as their next steps? And then they're going in and making more syntheses here for the following week. And then this is continuing to turn on a weekly basis. And typically our projects may here run for 12 to 18 months before we ultimately zoom in on the compound of interest. So we got a lucky break. We decided that we wanted to destroy the symmetry of our molecules. We thought our molecules here were a little bit too big to be good oral therapeutic agents. And so the team chopped off a portion of the molecule. And you'll see here going from 12829 through to this molecule, we decided we were going to take a bit of a hit on the potency of our molecules. We've clearly lost around 20-fold in terms of potency here. But what this did enable us to do, for the first time, this really enabled us to start getting molecules that had a little bit of oral bioavailability. So back squirreling away in the lab, they decided to make another change to the molecule, and now they changed out this relatively polar triazole heterocycle now for this pyridazine here. And not only did that give us back some of the potency that we had lost here, but it now really started to get our oral bioavailability where we wanted it to be. So this is the first indication that we're really heading in the right direction here. So back to the drawing board. Ultimately, the team probably made close to 1,000 molecules. It probably took the glutamate 1 team around a year and a half, 2 years before they actually came up to this molecule that's shown on the slide, which is IPN 690, the molecule that is now in the clinic. So it's really a case of triage up in this preclinical phase of drug development. So it's evaluating and really picking which is that molecule that we think is going to be the winner that we can advance into trials here. There's a lot of attrition, there's a lot of frustration, but every so often this gem will come along that you think has got all the attributes that you want, so it's going to be successful. So this molecule now is a really potent inhibitor of glutaminase 1 in vitro. In our biochemical assay, we're seeing an IC50, so we're inhibiting 50% of the activity of the enzyme, around 26 nanomolar here. The molecule is very nicely selective. It does not have any activity at all on glutaminase 2 here. So we've engineered in the selectivity that we wanted here. It's able to inhibit glutaminolysis here in cells. So I'm plotting out here the ratio of glutamine to glutamate. And in cells with an IC50 of around 14 nanomolar, we can really inhibit 50% of glutaminase activity. As we get up here to a couple of 100 nanomolar, we're inhibiting more than 90% of glutaminase activity in cells. And what we're able to see here is in our responding cell line, we're killing those cells here in the A549. But our non-responder, our H727, there's no phenotypical effects. So this is really, from an in vitro perspective, doing everything that we want. No need for all of you to read all of these numbers, but this is essentially all of our absorption, distribution, metabolism, elimination data, both in vitro in the upper half of the slide here, and in vivo as we start to study the molecule across our preclinical species. The PK in mice, the PK in rats, dogs, in humans. And from all of these numbers, we're able to extrapolate, and we're able to say that we think this molecule, it has good oral bioavailability in our preclinical species. We expect it's going to have good oral bioavailability in humans, and we're able to predict that this molecule probably is going to have about an 8-hour half-life in humans. That is probably going to be sufficient for once-a-day oral dosing, but most certainly it is going to be suitable for twice-a-day oral dosing. So this is encouraging, and it really told us that we're heading in the right direction. The other thing that we do is we dose increasing amounts of the compound to our preclinical species. And so we're dosing at 50, 100, 150, 200 milligrams per kilogram in mice, similarly increasing doses in rats, similarly increasing doses here in dog. And what we're able to see is a good increase in exposures. And so this shows that we really have a molecule here that's truly best in class. Other glutaminase inhibitors are clearly not able to show such nice pharmacokinetic properties here. So the team's engineered a really nice molecule here. And so when you start looking at the plasma concentrations here shown up on the y-axis over the duration of the 24-hour period, when we dose mice now with a dose of 250 milligrams per kilogram, we're able to see really high concentrations of the compound in circulation across that 24-hour period. Even if we factor in plasma protein binding here and looking at the free fraction, I mean, it's a free fraction of a drug that's important to inhibit a target, we're really able to inhibit the target here almost for the full duration of that 24-hour period. Most certainly, the black line shown above EC50 here, we're also kind of almost covering it at 10 times that EC50 here. So we've got really good confidence that this molecule is going to be able to inhibit glutaminolysis in human subjects here. So I think I should have been able to convince you here that we've got a good molecule here that's really going to be successful in patients. So let's really focus in on the disease-relevant biology and how we're going to use these molecules to treat patients. So in the background, the team led by Jeff here were really profiling the molecule across panels of cell lines in collaboration with investigators across the institution. And we were delighted to see that there were a subset of both non-small cell lung cell lines here and also ovarian cancer cell lines that really showed a strong dependency on glutaminolysis here. We were able to see killing of these cells in the couple of hundred nanomolar concentrations, whilst in gray there are a distinct set of both non-small cell lines and ovarian cancer cell lines that do not respond here. So when we started to profile those lung cell lines here and looking for their ability to consume glutamine and convert glutamine to glutamate, both our responders and our non-responders were able to show that IPN 690 effectively inhibited glutaminolysis there. But the true differences came when we started to look at global metabolomics profiling here. So in the black bars here, we can see changes across the subset of metabolites here in our responders, and in the open boxes here, you can see in our non-responders those H727 lines. So with IPN 690, you're seeing kind of a strong suppression of the TCA cycle. But you are seeing increases here both in purine metabolism, in the pentose phosphate pathway, but also the ribose salvage pathway. And there are differences here clearly between our responders and our non-responders here. So looking in a little bit more in our responder lines here, we're able to see a strong increase in the inhibition of glutathione levels in our responder lines here. So you can see treatment with around a micromolar of our glutaminase inhibitor, we're seeing a profound drop in glutathione levels. And ultimately, we know that glutathione is one of the main metabolites here responsible for maintaining redox. And treatment with our glutaminase 1 inhibitor here clearly gives us a strong induction of the active oxygen species that goes on to drive the phenotype in these responder lines. And we're not seeing this in our non-responders, in our H727 lines. So ultimately, going back and looking at those global metabolomic profilings, what we really hypothesized is going on here with our inhibitor is that we are seeing a global change in metabolic flux here. And so we're seeing a decrease in our responder lines here on TCA cycle, whilst we're getting a futile adaptive response here as we try to rewire cancer metabolism in our non-responders here around the pentose phosphate pathway and the ribose salvage pathway as we try to restore reducing equivalence in there. We're also seeing a rewiring of metabolism here, pushing in our responder lines some of the glutamine out there into nucleotides. So clearly we have a DNA damaging response going on here as well. So coming back and now profiling and looking at those responders here, and now the waterfall plots across our non-small cell lung cell lines here, we're seeing a distinct subset here of a responder lines. And when you start to going here and looking at what's special about the responding lines here, you will see a significant increase in both mutations in KEAP1 and NRF2 here. And as you probably are well aware that NRF2 here is a key driver of redox here, and really drives a redox program. And so in these responded lines here, there is a aberrant control of the redox pathway here. And this really gives us a hypothesis about where we want to take this program. We're interested in kind of taking our glutaminase 1 program into an NRF2 keep 1 mutant background here. So that's where we come to the next aspect. We've clearly demonstrated that we have an in vitro phenotype here. Now we want to kind of get into to study these in vivo. And the pharmacological audit trail is important. So how do we correlate the plasma concentrations of our molecule to their ability to engage targets, to show some changes in the fundamental biology, and ultimately here to just demonstrating a relevant phenotype. And again, the translation group has been able to do a really strong job here. We've demonstrated here that molecules can get to good high plasma concentrations of 100 milligrams per kilogram dose for a significant period of time here. And now in vivo, both in PBMCs and also in tumor, we're able to effectively inhibit glutaminolysis. And so looking directly at changes here upon dosage of IPN 690, we're able to see strong inhibition here of glutaminolysis, both in tumor, and we're able to see strong reduction now here in levels of glutathione in tumor biopsies. So this is good. Now, how do we kind of evolve this to really develop a clinical plan? And so now the in vivo team took this, work from Angela Harris and Ningping Feng, and together with several others in the in vivo group, really was able to demonstrate activity now in a setting of keep mutant non-small cell line, both cell lines and now in patient-derived samples here. Again, we were able to see in these keep 1 mutant models around 50% TGI, both in the cell line model and in the PDX here. When we get over on the right-hand side of the slide here to our keep wild types, we're not seeing any phenotype, really corroborating what we've seen there in vitro. And the reason this is important, if people recall back to Nando Scolitas' presentation from ASCO last year, is these keep mutant patients that really do not respond well in the clinic to immune checkpoint blockade. We know that these keep mutant patients are kind of poorly responsive to immune checkpoint blockade. They have worse PFS. They have worse overall survival here. So this is clearly an area of unmet medical need. So definitely worthwhile to take the project into this setting. So back to the ovarian setting now, and again, working with the Gyn-Onc group here, we were interested in understanding what was going on here. And very similar to what we've seen in the lung setting, we were seeing in the responders, so in the OPCAR8s, the SCOV3s, and in the 429 lines here, we're seeing a strong induction of ROS in the responder lines, and no real response here, no induction of ROS in the non-responders. Coming back and looking at the global metabolomics, in our responders, we're getting a similar level of changes to what was seen in the lung setting. The real difference now came in the non-responders, shown in the far right panel here in the blue. And overall, this was relatively benign. We really weren't seeing any changes. But what was dramatic was that there were some significant changes and significant consumption of both alanine and aspartate that are important for glutamate synthesis here. So looking at kind of how cells can get glutamate, they can essentially get glutamate through 3 different pathways. One is glutaminolysis, one is through this enzyme called ASNS, and the second way from GPT2. And when we started to look at our sensitive lines in comparison to our resistant lines, what was strikingly apparent was in the sensitive lines here, there's a very low levels of AS and S expression. So this really got us thinking. So we went back and we looked at our waterfall plot, and if you start looking now for AS and S low cell lines, you can see that these are the predominantly in our responders here. So we were able to go into a SCOP3 AS and S low cell line xenograft here, and are able to see some pretty encouraging tumor growth inhibition in this line here. And again, coming back to that pharmacological audit trail, show that we were able to strongly inhibit glutaminolysis here in those SCOP3 tumors. So again, in vivo team, in collaboration with groups on the outside, we're able to go into a subset of PDXs and looking at, on the left and in the center here, these AS and S low models, we were able to see some pretty striking phenotypes in monotherapy treatments here. And then working closely with the pathology group, we've been able to really refine these assays here and develop a clear-based assay that we're actually now using in the clinic for patient stratification. So in addition to monotherapy, we're also interested to start thinking about kind of adaptive responses and combination strategies. So leveraging some of the functional genomics capabilities that we have within the Traction platform, we're able to really conduct a functional genomics screen looking to see what happens when we include a treatment arm here of IPN 690. Can we see some strong synergistic activity happen here? So the team executed that study, and we were able to see a number of genes that dropped out from that screen, all related around the PI3K mTOR pathway, and gave us a strong hypothesis that if we were able to combine with agents such as an AKT inhibitor, an mTOR inhibitor, that we may be able to be seeing a stronger response here. So the team went along and conducted those screens. So monotherapy treatments with IPN 690 in red here shows around 40% CGI. The mTOR inhibitor, we're seeing about 80% CGI. But the combination here, we're seeing robust regression here, both in a cell line model here as well over on the right-hand panel of a PDX here. So really gave us strong confidence that a combination here with kind of the mTOR pathway or a PI3K based pathway here would be worthwhile and meritorious to do in the clinic. The team's got a wealth of additional data in the background, and in the interest of time, I'm not going to be able to speak about that today. But we've conducted additional metabolomic profiling. We've done proteomics analysis, looking at pathways of resistance and what else gets induced here. We've been doing some pretty significant immune profiling in the background, coming back to that tumor extrinsic activity. And all of this has helped us to refine now opportunities here. So I mentioned again previously the mTOR combination. We've been able to see some pretty synergistic activity with immune checkpoint blockade in a number of syngeneic models, and this is exciting for the clinic. And we've also been able to show, given that redox and ROS is important for detoxification of some of our standard of care agents like taxanes, we've been able to see strong synergy there with chemotherapy as well. So with all this data, it really encouraged us to push into the clinic. So we pulled together our IND-enabling package. We've clearly got a comprehensive preclinical package. We conducted all those GLP safety toxicology studies that the agency wants you to do. And we produced both drug substance and drug products to enable us to move into the clinic and really developed a defined clinical plan. But given there were so many opportunities in front of us here, we decided to engage a large pharma to help us carry this program most effectively into the clinic. And then we, back in May 2018, 2 years ago, almost to the day, we were able to form a joint partnership with Ipsen to take this program into the clinic. Ipsen rose the program from where we are. We're rapidly able to request the FDA to initiate first-in-human clinical trials. Ipsen got approval for that, and IPN 690 moved into the clinic. And that's where really the deep pockets of pharma has been able to come into play. And so this trial is now ongoing within the phase 1 unit led by Tim Yap. The first part of the trial here is really around monotherapy, study endpoints and what you would see for a typical phase 1 trial, safety, tolerability, demonstrate what the maximum tolerated dose are and the recommended dose, and then starting to understand the efficacy of this program. So preliminary anti-tumor activity, we wanted to establish a PK/PD relationship. We're interested in the biomarkers that we previously mentioned, and then look at some exploratory markers. Ipsen have been able to, are in the process of forming an alliance with Merck to access pembrolizumab for combinations here. We will be opening up a combination arm with taxane here, and really coming back to those responders that Jeff's team were able to identify. So those KEAP1, NRF2, KEAP lung patients, the AS&S low ovarian subjects, and the trial moves forward. Regrettably, Ipsen have not disclosed the response data, so I'm not able to tell you about that. But what I can tell you very briefly here is that we've got strong engagement of the target in patients. So collaboration from the translational group with the ADME PK team, we were able to develop an ex vivo assay, initially in rodents, but ultimately now in humans, whereby we can treat mice with IPN 690, we can collect blood, we can spike in label glutamine, we can harvest PBMCs, and we can really demonstrate that we're inhibiting glutaminolysis in those PBMCs. We've been able to show a strong dose dependency here in rodent blood. We've been able to show a really nice correlation across plasma concentrations and compound concentrations in tumor, and we've been able to show a strong correlation between inhibition of glutaminolysis in tumor versus PBMCs in this ex vivo assay. So really convincing us that this assay is by measuring what's going on in the plasma, we can tell what's going on in the tumor here. So this is the data from the phase 1 trial. So in the phase 1 trial, we've dosed our patients, we've collected the blood, and we're just spiking in glutamine, labeled glutamine here. And across the dose range in the phase 1 trial, we're able to see that we're seeing strong inhibition of glutaminolysis. And what is really encouraging here, as we're increasing the plasma concentrations with increasing doses of IPN 690 in patients, we are seeing really profound inhibition of glutaminolysis here. We're essentially titrating out this assay. And we're pretty convinced here at this stage that in human subjects here at MD Anderson, we are inhibiting glutaminolysis 24/7. We hope in the not too distant future to be able to tell you something about the patient responses here and how those NRF2, KEAP1, and AS&S low settings come in here. But the team has really been able to engineer a molecule that can inhibit its target effectively in patients. So in summary, hopefully I've convinced you that 690 is a best-in-class molecule. The translational team have really done a great job of dividing biomarker-defined populations. Collaborating with the pathology group, we've been able to put in place clear-based assays here, and those trials are in progress. And a huge thank you to the team in therapeutics discovery for making this happen. Whilst we're taking questions, I'm just going to leave this slide hanging there. It really speaks about that we have a call for proposals. We're looking for new projects that could come into the therapeutics discovery and development pipeline here. And really go online, please look at the application sites here. And would really love to collaborate with you both around the development of small molecules, biologics, and cellular therapies. And with that, I can take a couple of questions.
Thank you very much, Phil. It was a wonderful presentation about everything what means from start to the clinical work for drug development. It's very helpful for all of us. And let's start with the questions. How do you address tumor heterogeneity problem encountered in clinic? Cancer have some highly adaptable cells that can survive for a long time without glutamine in questions.
So I think that's clearly coming to the need here for us to develop these functional genomic screens to be looking at combinations, both combinations with other cancer metabolism targets here, but also with our immune therapies. I mean, it's really going to take a cocktail here to be able to address that that heterogeneity. I don't think, as you saw from that clinical plan, that any of us expect glutamine inhibition on its own to be truly effective. It's the combination that we need here.
Okay, so basically you are thinking through combinations between this drug and others.
I think we'll be able to see some clinical benefit as monotherapy. I think ultimately the use of these molecules in the clinic is going to be here, the combination.
Thank you. Another question. So for the GLS-1 inhibitor, what did the team think about the lack of correlation between biochemical and A549 cell potency early in the project? What assays ultimately drove the understanding of potency and selectivity?
So I think we have to remember that all of the assays that we develop are artificial. We use a fixed concentration of enzyme here in our biochemical assays. That is probably close to 10 nanomolar or so. And also, so I think part of it is in our in vitro assay, we're probably plateauing plateauing out the bottom of our assay. And then secondly, we don't really understand both the true concentration of glutaminase 1 in cells, how it may be post-translationally modified in cells here. I didn't show you the plot of biochemical versus cellular target engagement, but when you look at that plot, there's pretty good correlation, albeit with a shift between inhibition biochemically and inhibition in the cell. But as the requester rightly pointed out, our molecules do appear to look slightly more active in cells than they do in vitro.
Thank you. Yesterday, question from me. Yesterday was a very interesting talk by Kunar Rai about enhancers and all these, let's say, non-coding DNAs. Now, all these molecules developed till now, or mostly, are targeting the coding space of the genome. What is your opinion and what is your strategy for this very large 90+% of the genome not codifying for proteins? Do you think it's a targetable place, it's interesting for clinic and so
on? I think you're asking a really interesting question there, George, and I think what we're going to see with this decade, as this decade moves forward, is there are going to be molecules out there that are kind of targeting RNA. I think at this moment in time, we're really thinking about structural RNA, whether it be a ribo switch or something here that has a defined structure. But you're starting to see it now. You are starting to see people coming out and targeting RNA. So it's definitely an interesting space and something we're starting to pay some attention to.
Thank you. Another question. The mouse models only show the inhibitory effect of the drug, but not the regression effect. Is that a concern?
At the end of the day, all of these models are artificial. And again, as the previous question there came in, I mean, ultimately it's going to be a combination here.
Okay.
We really took our in vivo model here as a demonstration of activity. I mean, here we were seeing a meaningful phenotype. When you start to kind of go into a syngeneic model, you're seeing a stronger response there. you're adding on the addition here of the mTOR inhibitor, the taxane. It's the totality of the data here. Not all of our models here, when we grow a xenograft in the flank of a mice, that is a very artificial system here. We weren't ultimately concerned here because, again, the totality of the data in all of those key mutant models, in all of those AS and S low models, we're seeing kind of profound response on targets here.
Thank you. So basically, you are looking forward for the response in patients. This is what will say the truth for this molecule.
That's true. I mean, if you saw the number of plots on that PK/PD slide there, we've already got a pretty significant amount of patient information. Hopefully, in the second half of the year, we may actually be able to start disclosing some of that data.
Thank you. So thank you very much, Phil. It was a wonderful presentation. It was a big lesson for us how difficult and how exciting it is to develop these efficient molecules for patients. And with this, I want to tell you again, all of you, thank you very much. Big family of MD Anderson. You are for us, close to us for 6 weeks. And now from next week, we start a summer series of Friday, 10 a.m. Next speaker, Dr. Kati Rezvani, with a wonderful translational project which match very nicely with what Dr. Phil Jones presented today. And in fact, we are all happy. We are happy because we are going back to labs. We are going to make discoveries, because without the discoveries, it will be no drug to put in clinic. With this, thank you very much, and keep in mind, after rainy days come sunny days, closer and closer, and we will do what we love more and more. Enjoy science. Thank you, everybody, and see you next Friday, 10 a.m. for Kati Rezvani presentation. Good luck to all of you, and enjoy the weekend too.